So polysaccharides can be homopolysaccharides or heteropolysaccharides. Polysaccharides are even called as glycans, and homo means they are made up of same monomers repeated a number of times, and heteropolysaccharide means made up of more than one type of sugar. Then in the same manner, they can even be called as pentosan, hexosan, depending upon which type of monomer is there, pentose sugar or hexose sugar, or if arabinose is there, then araban. If xylose is there, remember this we have done, they are pentose sugar if you remember. So if xylose is there, xylan. In the same manner, we can have glucan or glucosan for glucose, fructan or fructosan for fructose, etc. Please remember all these things, that these are because of the carbon numbers, and this is because of which monomer exactly is there. So starch, glycogen and cellulose, they all are glucosan and hexosan and homopolysaccharide. Reason, they all are made up of glucose, so they are glucosan or glucan. And because glucose is hexose sugar, so they can also be called as hexosan. And because they all are made up of same sugar repeated number of times, they are called as homoglycan or homopolysaccharides. Then in the last thing which we learned was that functionally how many types of polysaccharides are there. So we learned that polysaccharides according to their function are grouped into storage polysaccharides, structural polysaccharides and the mucopolysaccharides. Now what are the storage polysaccharides? Those which exist as food. So starch, glycogen, they act as a food. And remember, I made a comparison between amylose and amylopectin. Amylose is not branched, but it is helically wound, and helical structure is more complex in the case of the amylose compared to the amylopectin. Amylopectin is branched, and I gave you the comparison between the number of residues or glucose molecules present. And because there are more helically bound structure in the amylose, that's why amylose is able to trap more iodine. So when a potassium iodide solution is used to perform a test, you will find blue-black color in amylose but a red-violet color in the case of amylopectin. Then I told the bond present is alpha 1,4, and if it is a branched one, it will even have alpha 1,6 bond also. Then glycogen is also called as the animal starch. It has 5,000 to 30,000 residues. It is just like amylopectin, but it has more number of branching, so the bonds formed will be again same. Then we learned that functionally the polysaccharides can have one more type, that is the structural or the fibrous, and reason they form the body or protective structure or the skeleton or the cell wall. So yes, we are talking about the cellulose. Cellulose example has a beta 1,4 bond. It's a linear arrangement. Remember, there are no branches present in the case of the cellulose, and it has 6,000 to 10,000 residues. It does not react with iodine because helix are absent. So there are no helix in the case of cellulose, but there are helix in the cases of the starch. And then I gave you the example of the enzyme which is present in the gut of many organisms, which is responsible for breakdown of cellulose. So beta glucosidase, most abundant and in the plant, and the second most I told was lignin to you in the plant kingdom. Then we learned that inulin is a homopolymer of fructose, so it can be called as a homopolymer. It can even be called as a hexosan. It can also be called as fructosan or fruxan. Reason, it is made up of fructose sugar. Remember, human body cannot digest it, so it comes out as such through the kidney, so it can be used to detect the problems with the kidney. Next is chitin. Chitin, I explained, is a heteropolysaccharide. Reason, it is made up of acetyl glucosamine. So this is glucosamine if there is an NH2 here, but because a side group, acetyl group has come, so it turns out to be acetyl glucosamine. Remember, there is a linear arrangement of the monomer, and remember, it has a bond formation of beta 1,4, just like in the case of the cellulose. Now the last thing we learned about was the mucopolysaccharides. That mucopolysaccharides provide slippery nature, and they can be They are of various type, like in the animal case I told you high hyaluronic acid. In the case of plants, joining the cells together is the pectin. Then in the collagen, you can have the collagen of the cartilage, you will have chondroitin sulfate. In the hair and all, you can have keratosulfate. And then in the case of red algae and green blue, in the red algae and the brown algae, agar, carrageenan, algin are there. So these all are slippery things, and they provide support, they provide help against desiccation, drying out, and they are used in the industries also, like in this case of soft tea, in the case of lamination, in the case of the cosmetics, polish making, mucopolysaccharides are used. Now, before we move on to lipids, let's quickly do some of the functions of the polysaccharides. So let's understand some of the functions of